Assalamu alaikum everyone. I'm Dr. Faiza, Director of Center to Advanced Level Research and Development. I welcome you all to this first international conference on research in biological sciences. Today I want to speak on importance of endometrial decedualization. I know this topic might be very new for few of you, but it's of a very big importance. I'm sure at the end of the presentation, most of you will be getting a new topic for your further research. So let's begin this. Before we can talk on importance of endometrial decedualization, let me first give you a short introduction about the movement of egg from the ovary to the uterus. I'm sure most of you are familiar with this process. It's just like after sexual intercourse, the sperm it swims through the cervix and the uterus into the oviduct where they meet an egg. Now one sperm, it may fertilize the egg to produce a zygote. You can see in the diagram, here the eggs move and then it covers this all path and reaches the uterus. You can see different days marked on it. This is how the egg moves from the first day till the 8th or 9th day when it implants in the blastocyst. Now the uterus has a thin spongy lining and the embryo sinks into it. Where this last step you can see this all. So this step is known as implantation. Now the egg when reaches into the uterus, it's implanting over here. It's implanting in the spongy lining. Now where's the spongy lining? This distance you can see over here. This is that spongy thin lining where the embryo has to implant. So this is the basic pathway of the egg, how it reaches to the uterus. But at the time when it's implanting, obviously there might be many changes which is taking place over here where the egg has to implant. The most important step which takes here is decedualization. Now in simple words, if you want to know that what is decedualization, so I must say that these changes when decedualization, the process is going on. Now these changes, they include recruitment of leukocytes and importantly the differentiation of ESCs. ESCs means endometrial stromal fibroblast cells into decidual cells. So decedualization, it refers to functional and morphological changes that occur within the endometrium, this part. So let's see this. Here you can easily see the cells have been differentiated and marked over here. I'm sure everyone looking at this cycle might have remembered that why we use this diagram specially. We always use this in our books for the menstrual cycle, so that you can understand different parts, different stages going on. Just like you see here, this is the shedding stage, then the regeneration, then decedualization. And decedualization involves the implantation window and the natural embryo selection window. So the process of decedualization for implantation is very important. And in which phase does this occurs? It occurs in the secretory phase. Now if you go through this whole diagram, you can see that during each cycle, the human endometrium, it undergoes a regenerative process. Now this regenerative process, it is characterized by the shedding, the proliferation and the differentiation. These all three steps takes place in the whole cycle. And upon decedualization, what happens that the human endometrium, it becomes receptive to implanting embryo. It means it's ready to implant the embryo. Now this period is implantation window. You can see over here. Now this is the process. This whole secretory phase, it involves decedualization process, and within this decedualization process you can see the implantation window. Sorry. Now this implantation window, it's a very short time when the egg, it has to implant into the endometrium. So this time is very important. If the decedualization process is going in a flow process without any damage, then the egg can easily implant. But if the decedualization process is not going in a flow, it has some damages, the cells are not properly stepped further, so the egg cannot implant. You can see the process over here in the implantation window. You can see it started to implant, and here where the natural selection window is taking place over here, within the natural embryo selection window, the egg, it implants into the endometrium. Now if you see the cells, which I'm too much saying that decedualization, what is decedualization, how you can differentiate it. Over here you can see pro-inflammatory ESC cells, they are in the red color, and the decedualized cells, they are in this, you can see over here, the yellowish color cells. So these cells, they begin to cover around the implanted egg, so they are very important. You can just say they are like shock absorbers. So it protects the embryo from it protects the embryo from ROS, reactive oxygen species, no matter how high it is, to the extent that it does not cross the threshold level. It protects embryo from a very high level of ROS. So these cells are very important for this. Now over here you can see the egg getting implanted. Once this implantation occurs, the embryo breaches the luminal epithelium, and the stromal cells surrounding the implanting embryo, they transforms into decidua. Now the decidual cells, they first are known as stromal cells. Once the egg is implanted, they started to change themselves and convert themselves into decidual cells. So when they are in the stromal cell form, they are like spindle shape. Over here you can see this ESC cells, the green shape. You cannot see these green cells further, right? It is because these green cells, now they have been converted to decidualized cells. That's why. Okay, so these are some of the experimental diagrams over here, which can show you that where is decidual cells present. Especially these cells, these diagrams are taken for the gene GPX3, which actually involves in the oxidative stress. So it protects from oxidative stress. So the diagrams, these diagrams which you're seeing here, these are ISH experiments, and the ISH, they performed to examine the spatial distribution of GPX3. Now these are the mRNA used in mouse uterus. So these are the days, the day 1, day 4, day 5, these are the night, and then the days, then day 6, then day 8. Now the black color, the brown, dark brown black color over here, this is showing GPX3. You can see how it is increasing day by day. So at day 8 it has a very high expression of GPX3, which means that these days, as the embryo is being saved by GPX3 from ROS species, so it keep on surrounding the embryo. Now when I say it keeps on surrounding the embryo, I mean this part over here, the embryo is present, the fetus might be growing over here, and these cells, the decidual cells, they are surrounding it. Now decidual cells, as the day passes, it also gets divided. means the secondary decidual cells, and then moves on. So to address that, whether GPX3 expression it was induced in the embryo, over here they examine the expression of GPX3 during pseudopregnancy as well. So pseudopregnancy experiment has to be involved in it, so that we can actually see that whether GPX3 expression was induced in the embryo or not, right? So these over here the diagrams, you can see this is the mice uterus showing how GPX3 is expressed in the decidual cells. Now next, over here you can see these are the controls, the pregnancy rate, and this is the MS. Now what are they showing over here? Now from day 1 to 5 of pseudopregnancy, there was no detectable GPX3 in the previous diagram we have seen it. So over here what the scientist has done, that the effect of mercaptosaccharic acid on the mice pregnancy. Why used this acid? Because it will be showing the effect of oxidative stress in it. It would affect it, especially this acid. It's a specific inhibitor for GPX. So we are studying the GPX3. That's why we use the inhibitor for GPX to see that what if GPX inhibitor is applied, and does it affects the pregnancy or not. So this inhibitor, it is injected by IP, and we use different dose for this. What the results were obtained. On the right you can see the control uterus of the mice and the embryos implanted in it. These are round balls over here, you can see very clearly. These are the embryos implanted, and this is the MS. This is also, here you can see very clearly no embryo implanted. This is very short and no proper embryo implanted. So this experiment showed very clearly that mice were sacrificed in the morning at the 8th day, and ovulation was confirmed by counting the number of corpus luteum, and the uteri over here, which has been photographed, you can see the very clear difference of implanted embryos, which means that if GPX inhibitor is induced in it, it affects the fertility of mice. So it's not only mice, maybe this GPX also act in the humans. So a lot of experiments are needed to move further. Maybe this all experiments, if done in the human, or like some miscarriages cases, or in this, GPX might be playing very important role in it. Again, these are some experiments for another gene known as FOXM1. You can see the diagrams, the colored area, the decidual cells surrounding over here, and the ISH experiment inflorescence, and the other showing the same thing. Here the uterus are shown very clearly. In the absence of pregnancy, the dark black arrows, the arrowheads. I hope this is very clear to all of you. That the arrowhead, the points which are over here, these are resorption sites, right? It means that the embryo has not been implanted very well. So these are the resorption sites. So the difference, this is the control and this is the knockouts. Now, what is importance of endometrial decedualization? As we have seen before, the importance of decedualization with the example of two different genes. over here I want to conclude with the example of humans, that what importance of endometrial decedualization, how you can study it. There are two basic diseases, disorders you can say, in human, the abnormalities, and those are endometriosis and chronic deciduitis. You can see the both of the pictures over here. These abnormalities in decedualization, they have been implicated in diseases such as endometriosis, which impaired decidualization. It leads to ectopic pregnancy, the ectopic uterine tissue growth. So the lack of decidualization, it has also been linked to higher rate of miscarriages. So it means that the miscarriages involves the impaired decidualization. The second one, over here you can see this dark brown colors. So this is chronic deciduitis, and it's a type of long lasting inflammation that arises in the pregnancy, and it affects the endometrial stromal tissue. It means endometrial stromal tissues, I mean the decidua. So it is associated with preterm labor, and this diagnosis test. Now the diagnosis primarily on the presence of plasma cells. So these brown color which you see over here, these are the plasma cells. Either the plasma cells are present in the cells, the decidual cells, or they are totally absent. So it's a chronic inflammation of decidua that linked with premature birth. So the premature birth and the miscarriages in human, they are directly linked with the impaired decidualization. A lot of work needed in this matter, but we need people to work on it. So thank you so much for listening this, and we are open for collaborations. If some of you think that you can work with us on this project, we are open for this, and we most welcome you. Thank you so much.